Hello, people. See if I can't give myself a different background, virtual or not. Let's go to Lake Iroquois today. There we are. This is me hovering over Vermont. I look like a ghost. My camera's terrible. Okay, can we start as usual by seeing each other's faces so we can just feel like we're in a class? Elijah. Elijah the prophet. I'm glad you're back, Elijah. I lost you there for a minute. Rocky, come on, you never put your face up. Everyone likes you. Except Beamalak. Beamalak doesn't like Rocky. Isn't that true? I'm trying to cause trouble in the class here. Stefan's ignoring us. Oh, there's Rocky. How you doing? And Vanessa right next door. At least on my screen. Nobody else wants to say hello to us. There's Nishi. Hey. Here's Gus. Hey, Gus, say hello. Gus, put your head up. Put your head up. Come on, get in the camera. Anyway, Gus showed up. He always does. He's got very little to say about developmental biology, though, I'm afraid. All right. All right, shall we go ahead and rock on this? I didn't grade your exams yet. I have to do cell biology first because they were Thursday, so I'm going to do that this afternoon, I hope, and then hopefully get to you guys tomorrow. Hopefully by Wednesday they'll be done, but fingers crossed. We'll see how things go. All right, so let's go ahead and start in. I want to review what we talked about last Monday because it was a while ago, and we're doing this new chapter, so chapter 4. So chapter 4 is basically going to go through all the same Xenopus development that we did before, except now we're going to see why it actually happens. What are the molecular determinants of the of different tissues and how do they get induced? So, that in mind, fate mapping. I'll talk about this more later, but even at the early stages, you can determine what part of the embryo is going to be what in the final product based on where it is and where the sperm entered. And again, going way back, we've got at the egg stage, you've got an animal pole and a vegetal pole, and there are different tissue-specific mRNAs that are attached to the cytoskeleton in each of those ends. And so the brown end, not that the brown has anything to do with it, but the brown end will always be the ectoderm, and the yellow part, the yolk, will always be the endoderm. After fertilization and you get your first cleavage, you've got two separate you've got two cells here. If you separate those, you'll get two complete embryos. I've got a picture of them later. They're a little bit smaller than a normal embryo, but they're perfectly normal and they'll grow up to be healthy, happy adults. At the 4-cell stage, you can't do that anymore. Okay, if we assume that the sperm came in on this side over here, then these two cells will be dorsalized, okay, because dorsal determining factors are already set up at this end. So that means these two cells will be ventralized. Now, after the third cleavage, which comes across the middle, now we clearly have two different tissues here. And two of the three tissues we need to make an embryo. We've got ectoderm on the top, which is the animal cap or the animal pole, and then the endoderm on the bottom. And here's what happens if you cut them across at that 4-cell stage and you separate the dorsal determinants. You end up with the ventral side, these guys turning into just kind of amorphous ventral tissue. And the dorsal half kind of goes ahead and makes itself an axis, but it's missing gut. So this will not develop in the end. It's a normal one. This is so basically this will give you your whole brain, spine, all of that bit, but there's no gut, so it doesn't develop any further. Okay. Oh, here it is, the twinning experiment. I guess I should put this earlier. But if you cut right down this way, after that first division, separate them, you get two normal embryo, more or less normal embryos. They've got all the structures they need. They're just a little bit smaller because they started with half as much cell material. Same genetics. So they'd be considered clones of each other, genetically identical. Okay, so why is this happening? Now we're going to get into molecular stuff. And that's because you've got messenger RNAs that are restricted to different parts of the embryo. This is spelled wrong, of course. Vegetally restricted factors. So on the bottom here, you've got some really important endoderm specific factors, VEG1 and VEG-T. VEG1 is a hormone. It can diffuse a little bit. VEG-T is a transcription factor. So we haven't talked about cell biology much, but typically the way it works is the hormone binds to a receptor, and then that starts a signals transduction pathway, and somewhere down the pathway you activate a transcription factor, and that goes into the nucleus and changes which genes are turned on. Now, the type of tissue that the cell is determined by what genes are turned on, right? Because every cell in the body has got the exact same genes, and what makes one tissue different from another is which of those genes are actually turned on. So transcription factors and hormones that turn on transcription factors are going to be really important in this chapter because we're changing cell specification. We're going from a general cell into a more and more specific cell, which means turning on more and more genes that are specific for that final product. Okay, so vegetally restricted mRNAs include these two that'll make it vegetal endoderm. And then we've got these three, which are going to be dorsal specification markers. So beta-catenin, I'll talk about today what that is. Wnt11, so beta-catenin is this transcription factor. Wnt11 is the growth factor that causes beta-catenin to increase in the cell. And disheveled is part of the signal transduction pathway of Wnt that produces the increase in beta-catenin. So all of these dorsalizing factors are focusing on beta-catenin. Increasing beta-catenin will cause a dorsal specification. Now at the maternal egg, at the egg stage, these dorsalizing factors are at the bottom of the vegetal pole. So they're sitting square on the bottom. And that's because at this point, we don't have any axis. We've got one axis, animal, vegetal, but there's no other axis. When the sperm hits, we're going to see a change in the cell based on its cell cortex moving. So cell cortex is a mesh of actin microfilaments that surrounds the whole outside of the cell. Now this is a pretty bad figure because this is a intestinal cell and it's got these microvilli sticking up. So this is not what we're interested in, but I just wanted to show you what the cell cortex was. And here's immunocytochemistry on the cell cortex. You can see this red is that actin cytocortex or actin cortex all around the cell. Okay, so our egg also has this cortex all around the outside. Mr. Sperm comes along and it can only enter in the animal pole. So it enters somewhere in the animal pole. And as a result of that entry, this cortex then rotates away from the sperm. And so you get these dorsalizing factors attached to the cytoskeleton that then rotate up and into this kind of marginal zone, more or less, somewhere between the animal and vegetal section. Now we haven't divided yet. This is still just a single cell embryo, so this can move without any being impeded by any plasma membranes. So now that's going to be, it's not an axis yet, but it's setting up basically all three axes at this point, because this is going to be dorsal, that's going to be ventral, this will be actually posterior and anterior isn't really set up yet. But anyway, eventually this will become our dorsal end because of those factors. Okay, so now we're going to get into what those factors are actually doing. So here you've got these factors, the three I already talked about, disheveled, Wnt11, and beta-catenin, and a third one, GSK3 binding protein, which is going to be also involved in the signal transduction pathway. And then in the ventral side, I'm sorry, I'm a stickler for my things looking perfect. Down here in the endodermal side, we're going to see this protein, I wouldn't call it, I guess it's a signal transduction protein, being expressed down there. And this is going to stop any of this Wnt signaling. So the Wnt signaling will only be here and only make this dorsalizing, and the Dickoff will stop the rest of the place from becoming dorsal. Okay, so this is a new material. I believe I went that far and stopped last time. If not, you can chat me a question there. But everything here is focusing on this Wnt pathway. So what is the Wnt pathway? If you've had cell biology with me, we went over this, but I know most of you haven't. So the Wnt pathway, like when there is no hormone around. So this is like the Wnt pathway shut off. There's no hormone here. These are the receptors here, frizzled and LRP56. Here's our disheveled that we talked about before. It's in the pathway, but with no Wnt, it's inactive. And then here's the active part. This is what's important right now. We've got this complex we call the destruction complex. And it is composed of APC, which is adenypolyposis coli. When this is lacking, you get colon cancer. So that's why it's named APC. And then you've got axin, CK1, and GSK3 beta. Now, the business end of this whole thing is GSK3 beta. It stands for glycogen synthase kinase. So in the insulin pathway, GSK3 will phosphorylate glycogen synthase, and it will cause an inhibition of glycogen production. Not important for this class. We talk about that in cell biology, but it has a really important signal transduction role, as well as to, energy control role thing. GSK3 beta will phosphorylate beta-catenin. Beta-catenin is the transcription factor. So when GSK3 beta phosphorylates beta-catenin, it makes it a target for ubiquitinating enzymes. So here's another thing we haven't talked about yet, but it's really important. And that's like when a cell wants to get rid of one specific protein for some reason, it will ubiquitinate it, which means just putting little proteins, ubiquitin proteins on it, chains of them. And that causes that protein to go to a structure in the cell called a proteasome. And the proteasome will then take that ubiquitinated protein, take the ubiquitins off so they can be recycled, and then degrade it so it gets destroyed. I think I'll put if I haven't already, I will put a video up about this process. It's really neat. It's not vital to know it for this class. Just know that when this beta-catenin gets ubiquitinated, it gets destroyed. Okay, that's what's most important. But the way it's done is through the ubiquitination pathway. Okay, so why is that important? Well, in the cell at rest, the cell's constantly making beta-catenin, okay? Constantly making it, and it's constantly being destroyed because of GSK3 beta. So it's being made, but it has no effect. But then down here, you can see we've got some transcription factors that are bound to the regulatory unit of this gene. So here's a gene, something that will get turned on by beta-catenin. And you've got TCF, which is also called LF, and a couple other ones. And this bad boy Groucho here. So Groucho, again, named from Drosophila. I think we talked about that in this class. Groucho is a protein that is a repressor. It binds to TCF and it blocks transcription from all of these genes that are beta-catenin sensitive. Okay, so you got Groucho, TCF, and beta-catenin. Right now, nothing's happening because there's no Wnt around. Beta-catenin's being made and destroyed. Its level stays really low in the cell. Now you've got Wnt around. In our example, is Wnt 11. Wnt is a family of genes making different forms of the same hormone. So these hormones all go through a pathway like this, but the receptors are specific, so you can get different geographical expressions of it. But anyway, that's not important. What's important is when Wnt is around, it will bind to its receptor, which is called frizzled. I don't know if I said that up here. The Wnt receptor is called frizzled. Again, Drosophila. They mutated a whole bunch of Drosophila. Some of them look kind of frizzled. Some of them look disheveled. So anyway, Wnt binds to frizzled, and then that binds to LRP, and that makes a receptor complex. Now that receptor complex will recruit disheveled. I don't see disheveled here for some reason. It should be. Disheveled should come in first, and after disheveled, these other proteins come in, axin, APC, and GSK3 beta, K, and K1, okay? So that destroys the destruction complex, or breaks up the destruction complex. So GSK3 beta now becomes inactive. And because GSK3 beta is inactive, we're no longer phosphorylating beta-catenin, we're no longer breaking it down, and it builds up in the cell and binds to TCF, and then you get transcription from a whole range of genes that are going to be important for dorsalizing this end of the embryo. Okay, so let me just review that a little bit faster with a little less detail. Bottom line, we're bottom lining here. With no Wnt around, the receptor and mechanism is shut off. And you've got the destruction complex that's constantly breaking down beta-catenin as it's made. When the hormone's around, Wnt, it sets up the receptor complex, which then breaks up the destruction complex. Beta-catenin builds up in the cell, and then you get transcription from those genes. Okay, so that's what's going to be our dorsalizing signal. And why is it dorsalizing? We'll see here. Here's all your dorsalizing factors. And their beta-catenin is going to induce the transcription of a protein called siamois. Let me write that because it should be here and it's not. New-Coupe center. And it is made by siamois, which is a transcription factor. Okay, so beta-catenin induces the transcription of siamois, which is another transcription factor, and siamois will then go on to cause transcription of other things we'll see later in this chapter. And that makes these cells here the New-Coupe center. Now, that's not the organizer yet, but it produces an organizer. So this is our first step towards getting an axis, is making this New-Coupe center. And how do we know it's an organizer or organizer inducing? Because of experiments like this. This is a twinning experiment. So you take two embryos. This one, we're going to pull out this cell at the 32 cell stage, and it's where all those dorsalizing factors were. And we're going to open up this other embryo and stick it in there. So now you've got two of these cells at this 32 cell stage. Now you've got a 33 cell stage embryo, and they both induce, each of them induces its own axis. Okay, so you end up with this twinned embryo. Which is an abomination before God. It's pretty gross. It's amazing what can happen in embryology when you change the computer program and know what's going to turn out. So anyway, this is how we know. The whole point of this experiment is this is how we know New-Coupe center is important for inducing an axis, because if you take it out of one embryo and stick it in another embryo, you get another axis. By the way, these cells that were taken were endodermal cells, right? Put them over here amongst the other endodermal cells. And in the end, if you take these and dye them and follow where they go, they stay endoderm. So it isn't like you took dorsal tissue from one place and put it here and it's still dorsal. No, it's still ventral, or not ventral, it's still endodermal tissue, but it's able to induce other cells to become dorsal. And that's key. That's what this chapter is all about. How does one cell make another cell change? Okay. What do we got next? Okay. So fate mapping. So now we're going to talk about how these different tissue interactions produce the fates in these tissues that we see. So we talked about this earlier, fate mapping, in terms of you have endoderm at the bottom. That will be gut. At the top, you've got ectoderm. Now this ectoderm at this side will become neural tissue, so we call it neuroectoderm. And the ectoderm on this side, the ventral side, will all be skin. So that's epidermal ectoderm. The band in the middle at the marginal zone between endo and ecto is all mesoderm. And even at this early stage, we know the mesoderm over here is going to be more dorsal. It's going to be notochord. If you remember from Neurulation chapter 3, notochord is the most dorsal endodermal tissue, or I'm sorry, mesodermal tissue. Above that is just ectoderm. So this is the most dorsal mesodermal tissue. Over here is ventral mesoderm, things that'll come out of the front of you, your heart, your circulatory system, your kidneys. That all starts from this ventral as endoderm. And in the middle is the middle mesoderm. I think in the notes I call it medial mesoderm. This will be the somites, which then becomes the body cavity, the arms and the legs, and all of the that part of your body. And I think it's kind of neat to look at these things and try and imagine how this is actually happening. And the reason why I think it's not necessarily neat, but important to do this on a regular basis is taking these two-dimensional pictures and trying to imagine what's going on in 3 dimensions is really hard. So the more you do it, the more it makes sense. I've been teaching this for the love of God, 6 or 8 years anyway. And every time I look at this, I learn something new. Something makes a little more sense than it did last time. So at this stage, say you're at the blastula stage, this is all normal ectoderm. It's all just general ectoderm, pluripotent ectoderm. And right here is your dorsal lip, the blastopore lip. And these mesodermal cells, which are general mesoderm, they're not specified dorsal yet. And these endodermal cells are going to come in through that dorsal lip. And as they do, they get induced to be dorsal mesoderm, and they crawl across the inside of that ectoderm, and they induce this ectoderm to be neural ectoderm. And at the same time, this other ectoderm is just dividing and covering the cell with that epibole action that's going on. So, oops, didn't do that. So as these tissues here go in and crawl across at the top of the vault and they elongate the embryo, they're coming in this way. And you can see there's your most dorsal mesoderm, which is your notochord. Underneath that is the somites, and then you've got the more ventral mesoderm underneath it. Okay, that's where our layers come from. Now, I forget why exactly we went through all that, but I think it's worth talking about anyway. Let's move on. Okay. So how do we know this? This is where they're fated to be. How do we know that there's actually cell-cell communication that's causing all this to happen? It's through these experiments called explant experiments, where you take an embryo, you cut it apart, and you take different tissues, and you find out what is it specified to be at that particular moment in time. So this is your blastula. I used to do these experiments too. What you do is you take an eyelash, take it, and then you get a stick with a little bit of wax on it. You dip it in hot wax, you put the wax, you put your eyelash on the wax, and that's your scalpel because this embryo is so small. You're there with an eyelash cutting it apart. So what you do then is you take this embryo, stage 8. It takes about maybe 4 hours for the fertilized embryos to get here. And then you cut that whole cap off. Now, it's really important that you're taking just the cap and you don't want any of this marginal zone. You take that animal cap off and you culture it in, culture medium. It's epidermal cells. Okay, you get no neural tissue. All you get is epidermal cells. You take this vegetal section, again, getting rid of the marginal zone, and they stay endoderm. Okay, so at this stage, you can say these are not yet fated to become neural tissue, and there's nothing here is fated to be mesoderm, nothing from the top or the bottom anyway. So this is how we know that it isn't like maternal RNAs put down, turn this into neural ectoderm. That neuro induction is going to happen later. So that's what this experiment shows us. Now, the next experiment, we're going to look just at the medial, or mainly at the medial anyway. So here's another stage 8 embryo. We're going to take the animal cap off, and that becomes skin, epidermis. And then we'll take the marginal zone out. And if you take the marginal zone where the blastopore lip is and let it go, it becomes dorsal mesoderm. Okay. And you also get some neural tube because of the ectoderm that you had there. If you take marginal zone from this side, you get ventral mesoderm, blood vessels, things like that, as well as epidermis, no neural tissue, just epidermis, which covers everything. And again, if you take the endodermal cells, they just endodermal cells, they just stay endoderm. So what does this tell us? So unlike the last one, this is already specified. This has been specified as dorsal. So where that blastopore lip forms is already specified as dorsal. Why is it? Because of that cortical rotation we talked about, right? That happened at the 1-cell embryo stage. So those beta-catenin inducing proteins are already here. So this produces beta-catenin and it turns into this. But historically, at this point, they don't know that, right? So if we look at these experiments in terms of when they were done, they were probably done before anybody knew that DNA was a genetic material, early 20th century. So what they showed there was that these animal cap cells are fated to be ectoderm. This is going to be dorsal. This is going to be ventral. Why is that? Why would that be? I ask you. Well, the next experiment starts to give us a clue why that is. So here, what we've done is taken out all the marginal zone. So we know the marginal zone is specified to become dorsal and ventral mesoderm, right? So we take all that out. So we've got tissue that's only going to be ectoderm and tissue that's only going to be endoderm. And we put them on top of each other. And what happens is some of these ectodermal cells become mesodermal, okay? That tells us that there's some kind of a factor or factors being released by the endoderm that is changing the cell fate of the ectodermal cells, making them become mesodermal, okay? So this is the first experiment that showed that cell-cell communication is causing this specification, causing this induction of different tissues. So this shows you that endoderm, there's something being released from endoderm that's turning ectoderm into mesoderm. This is the point where you all cheer, and if we were in class, you'd pick me up on your shoulders and you'd carry me out and you'd be like, "Ah, you taught us something, Dr. Slish, yay, this is exciting." Unfortunately, we're on Zoom. Okay. So any questions about that? I know I went through a lot. I feel like I'm going too fast when I'm in Zoom because I'm not getting any feedback from anybody. Let me stop this share for a second. Anybody have a question about that? Vanessa is saying no. Nothing? You got nothing for me, eh? Let me look through my notes real quick and see if I skipped anything. Oh, there's a chat. Tyler Montano says nope. But Tyler Montano's very smart, though. He understands things easily, if you don't mind my saying. Okay, organizing centers, new coop, fate mapping. My notes are up there too, by the way, which probably explain more than what I've been talking about, or explain different things anyway. Okay, explant experiments, animal caps. Okay. So, vamos a continuar. Shall we keep going? Yes, Dr. Slish, let's keep going. Okay. You know, it's funny. Every time I look at this full screen, I see Akiela Simon smiling at me, and I'm like, hi, Akiela. Oh, it's just a picture. Never mind. I feel like she's always smiling at me. Makes me feel good. Don't take it down, please. Okay, well, what are we doing? What is this class? Oh, this is developmental biology. Okay. So what is this signal then that's inducing ectoderm to become mesoderm? Well, as it turns out, there are a number of different hormones, but they all use a similar pathway. And the pathway is called the TGF beta signaling pathway. So it's called this because the first hormone that was found to use a pathway like this was called, transforming growth factor beta. There's transforming growth factor alpha, which you never hear about anymore because I think they named it something different afterward. I think I don't know, let's not make something up. So TGF beta then is a hormone that's used on a lot of different things during development especially, but even after development. It's important in wound healing, and it can either or both turn genes on or turn genes off. So it's a really important signaling transduction pathway. We talk a lot about it in biology of cancer, which, if I could put a plug in, if you like signal transduction, that's a really good class for signal transduction. And I'll be offering that next fall. Anyway, TGF beta signaling. The way this signaling works is you've got two different receptors for the hormone. You've got a type 1 receptor and a type 2 receptor. Typically, they are kind of attached to each other, but when you add the hormone, it doesn't show it in this figure, but when you add the hormone, you get like two type 1s and two type 2s together into a big accumulated mass. And this is a classic signal transduction mechanism called recruitment. So when the hormone's around, it recruits all these receptors together. They make a big accumulated mass, and that turns them on. When they're turned on, the type 1 receptor phosphorylates the type 2 receptor, and then the type 2 receptor phosphorylates the type 1 receptor. These receptors are kinases as well as receptors. So once they phosphorylate each other, they become even more active as kinases. And then they phosphorylate something called a SMAD. So this first SMAD that interacts with the receptor is called a receptor SMAD. Okay, SMAD. I think SMAD stands for something mothers of decaplegic, something like that. It's a weird name, another Drosophila associated name where Mothers Against Decaplegic was was the name of a signaling molecule. So SMAD then becomes something related to Mothers Against Decaplegic. Again, don't worry about the name. Just remember SMAD. So the receptor will then activate the receptor SMAD by phosphorylating it. And then the receptor SMAD will then bind to the cytosolic SMAD that's sitting around, and that'll form a big SMAD complex. And that SMAD complex then will go into the nucleus and activate genes. Okay, so now you're getting specification because you're turning different genes on. Different genes produce different proteins, and you change the type of cell that is. So what's important to note here for our Xenopus differentiation is we've got two different types of TGF-beta-like hormones. We've got BMP4, which we'll see a lot, and then XNR. XNR is Xenopus nodal related. Nodal is another Drosophila protein, which is a TGF-beta-like hormone. And these are the Xenopus forms of the nodal protein. So XNR is Xenopus nodal related. And I put an S on the end of it because there are, I believe, 4 different XNRs, XNR 1, 2, 3, and 4. And I've got a chat question. Let's see if I can answer it. I'm going to stop my share first. How are these two receptors different? Good question. So the way they're different is if they come from different genes, so they've got different amino acids. And the amino acids in this type 1 and type 2 receptor only bind BMP4 and not XNRs. And this one only binds XNRs and not BMP4s. And they're also different at this end because this is only going to interact with SMADs 1, 5, and 8. And this is only going to interact with SMADs 2 and 3. Okay, so that's how they're different. They're coming from different genes and they're different proteins. So what we'll see is during mesoderm induction, we've got BMP4 coming from the ventral side everywhere from the endoderm, and then XNRs as well. XNRs 1, 2, and 5, I believe. 1, 2, and 4. And then later we'll see XNR3 coming into play. Doesn't matter. We'll go into detail later on it. But the bottom line is you've got basically 4 different hormones coming from the endoderm that are going to interact with the ectoderm and change its cell fate. You've got BMP4, which is ventralizing, and XNRs 1, 2, and 4 that are more dorsalizing. And the difference is which receptor SMAD they activate, and therefore what genes they activate. Okay. Take a quick look. Okay, any questions about that? Comments? We still have 12 minutes left in this class, and you people paid for this 12 minutes. You bought my time for the next 12 minutes. So I really don't have anything else to talk about. Do you guys have any questions or comments? Again, typically I'm going a little bit faster here because I can't interact with you people like I would otherwise. Any questions? Anyone want to chat a question here? Nobody? No one's got the guts to ask a question. Or was that presentation just so clear that there were no questions left to ask? If you don't have any questions, what I would ask is that you write an email to the dean and tell her that Dr. Slish is amazing and he deserves a raise. Not a lot, $10,000, that would be enough. Would make things better. But no questions, no comments. All right, well, why don't we call it a day? Are you sharing? Absolutely. It's called a kickback, Tyler. You get me 10 grand, I get you 500 apiece. How's that? We good? 10K isn't a lot? 10K is okay. Say less. Okay, guys, we'll see you Wednesday, and hopefully I'll have your exams at least close to done by then. Talk to you later.